Hey ARA community, Mr. Cooper here, your head of biology, here to talk to you today about the coronavirus tests we're doing. So, to give them their full name, they're called colloidal gold-based immunochromatographic tests. So that's quite a mouthful, but a couple of interesting things. They're super fast and not too expensive, which is great for us. But also, as the name suggests, they actually have gold inside them. Now, these are just nanoparticles, so they're very, very small, but it's certainly interesting to note. And I've got those represented on my piece of paper here by these big red gold circles. After all, it is these gold nanoparticles that are responsible for creating these red lines that we read when we've done the test. So they are actually negative in charge, which means it's really easy to stick them to these Y-shaped proteins called antibodies. Now, antibodies have an important role in our immune system, and they help us combat disease. What they do, what they're really good at, is they're good at sticking to parts of the virus. And as you can see, I've got these represented here with these horrible little faces to show that they are the virus. And so you can see that if the part of the virus has the right shape, it can actually stick to the antibody. And that's basically the main mechanism, the main way this test works. So I've actually cracked one of these open so you can see inside, and there's basically different parts to it. There's the sample pad. There's another pad just next to it which has all of these gold nanoparticles and antibodies in. We then have the test area. So there's a T part and a C part. So a test part and a control part. And then you have this absorbent pad at the end. And that's responsible for drawing your sample all the way across the test zone so we can have that result generated there. Now, if someone is unfortunately suffering from coronavirus, it's very likely that in their body they will have these virus parts. Now, we call these antigens, and what these antigens can do is stick to the antibodies that are attached to these gold nanoparticles. Now, as the sample is drawn through the test, what happens is it meets a couple of stages. So, as we discussed, we have the test area and the control area. So I've represented that here. We have a test area and our control area. So let's illustrate the gold antibody making its way across the test here, and what it's actually going to do is it's going to stick to these other antibodies here as part of this test zone. As you can see, it's the perfect shape, and if you imagine lots of this happening, you can see why we'd get such a nice bright red line here. Now, we also importantly have the antibody and gold molecules moving through the test, and what can happen as I lift this over to the other side is there's also a control region that has antibodies that actually stick to the antibodies on the gold region, and this actually lets us know that the test is working correctly. And also, because we've had some red lines occurring in the test region, this is someone who is positive for coronavirus. Finally, in someone who doesn't have coronavirus, we don't see the virus parts binding or joining to these antibody regions connected to the gold particles. So what happens is they are drawn through the test area, but they don't bind to this test region. Instead, they bind straight away to those antibodies in the control area, and we just get that single line in the C zone. So that shows the test is working, but because we have an absence of red markers in the test area, this person is highly likely to be negative. And I guess that's what we're all hoping for until the end of the year. So best of luck with your testing, everyone, and let me know in school how it's going.